Hello and welcome to the HIV Cure podcast. My name is Sakabo Sasalamarumo, and we are back again with another episode. And today we're talking about breakthroughs in HIV cure research. And I don't know, earlier this year we did see a lot of waves going on about an HIV cure being found in South Africa and all of those things. So there's no better person to come and have a conversation with us when it comes to breakthroughs in HIV cure. And today I have Professor Thumbi. Hi.
Hello, and thank you for having me. It's a pleasure to be on.
Yes, thank you for making the time. And when I was having a conversation with Professor Catherine from Botswana, and when we talked about it, she was like, "No, I know the perfect person, and Thumbi is gonna do this." And I'm so glad I had the opportunity to meet you during the conference, the IAS conference in Kigali, and then we had a conversation, and here we are today.
Yeah, no, it was a pleasure, and it was a pleasure to meet you as well during the IAS meeting. It was very inspirational, the talk that you gave at IAS. So, yeah, looking forward to this discussion.
Yes, thank you. So before we dive into it, can you kindly just introduce yourself and the work that you do for those who might not know you, so that they get to appreciate? And yes.
Yeah, okay, thank you very much, Sabo. I'm Professor Ndung'u. I work at the Africa Health Research Institute in Durban, South Africa. I'm responsible for basic and translational science at ARI, at the Africa Health Research Institute. So mostly we work a lot with the patient or participant samples. I'm a virologist by training. I come from Kenya, and my interest really is in how the immune system fights HIV and how HIV is able to overcome the immune system. Because that's what happens most of the times when the person is not on antiretroviral therapy, the virus usually wins. And so that's what I do. I study how the HIV virus interacts with the immune system. And the goal of my work is really to develop either a vaccine that can prevent HIV or a cure that is based on having the immune system suppressing the virus to undetectable levels so that the person can live with the virus to experience disease progression. So that's what I do.
Oh wow, you do amazing work. And what I also love is how you touched on a little bit about vaccines also, which we will talk about later on. And to our listeners, we did also just talk a little bit about the vaccines. If you guys want to, you can just go back to, I believe it was episode 6, where we're just talking about HIV vaccines in terms of research. So you can go and listen to that. And today we're talking about breakthroughs in HIV cure research. So can you kindly explain what breakthrough means when it comes to HIV cure research, and also just give us examples of some of those breakthroughs?
Yeah, I think the word breakthrough can mean different things to different people, but I like to see a breakthrough as a significant advance. It's when there is a significant progress, like a step forward that has been made in the area of us moving towards a cure. So when there is new scientific knowledge that moves us forward, I can call that a breakthrough. And one such breakthrough that we experienced more than 15 years ago now was the cure of HIV for somebody called Timothy Ray Brown. He was the first person, as far as we know, who was cured of HIV. He had to undergo a bone marrow transplant, and the bone marrow transplant, which is a very complicated procedure that he got. He got donor cells from somebody who does not express the CCR5 proteins on their cells. And now the CCR5 protein is a protein that HIV uses to get into cells. And so because he got these donor cells from somebody who did not express CCR5, Timothy Brown was cured of HIV. And a lot of research was done on him, and they could not detect any HIV in his body. So that's a breakthrough. That's a big breakthrough. Another breakthrough I can talk about is our own study in the fresh cohort, where we have given women broadly neutralizing antibodies and then asked them to interrupt treatment. And we saw that a few of those women, or 20% of them, through HIV, drugs for more than 1 year. So because this is the first time this kind of work has been done in Africa, and it shows some promise, although not entirely successful, I can say that that's also a breakthrough. So yeah, that's what a breakthrough is. It's just when there is a significant advance that energizes us and excites us about the ultimate goal, which is to cure HIV.
And you did give an example of Ray Brown, the bedding patient, and these are, like you did say, that they're one of the first breakthroughs. So I just want to know that with these first cases, the Berlin and London patients, and yeah, can you just also explain? You did explain the Berlin, and then please explain the London patient, and like who these were and how did they inspire hope for an HIV cure, and also the most recent studies that we've been seeing that have sort of like, yes, got information or were inspired through the first cases of HIV cure breakthroughs that we have experienced so far.
Okay, so the Berlin patient is actually Timothy Ray Brown. It's the one that I've just explained, where Timothy Ray Brown was a gentleman who was living with HIV. He developed a cancer, and the cancer required that he should get a bone marrow transplant in order to treat the cancer. Otherwise, he was going to die. And the bone marrow transplant, the doctors decided that they should give him a bone marrow transplant. What they do is they destroy the cells that you have, your own immune cells, and they give you a donor cells, immune cells. And the idea is, because your own cells might have cancer, the idea is to remove your own cells and replace them with donor cells. In the case of Timothy Ray Brown, he had been living with HIV. He had been on antiretroviral therapy, so his viral load was undetectable. So when he got the cancer, they decided to do a bone marrow transplant, but the donor cells were coming from somebody who does not express the CCR5 molecule, which is the molecule that HIV uses to get into cells. So there are some people who are genetically, they are made that way, that they don't express CCR5, that HIV uses to get into cells. So because he got donor cells from somebody who did not express CCR5, the virus could no longer replicate in his body. And when they tried to look for HIV within his immune cells after he got the bone marrow transplant, they could not detect any HIV, even though now he was not taking antiretroviral medication. And they were able to follow him for more than 10 years, and they could not detect any HIV able to replicate in his body. So that's why we call it a cure. And the same case applies to the London patient. So very similar procedure, maybe a few differences here and there, but the same case is somebody who developed cancer, he had to undergo a bone marrow transplant, and got donor cells, and then was able to stop antiretroviral treatment, and HIV was not able to come back. So that's a huge breakthrough, and there have been many now cases like that where that have been repeated, where people got bone marrow transplants without the HIV coming back. Now that's a very expensive procedure, so it's not something that can be done for every single person living with HIV, and that's why you don't see scientists or clinicians doing it on a regular basis, because the bone marrow transplant itself is not a very safe procedure. But it is a very important development and advance in the field, because it shows that people can be cured of HIV.
Okay. And earlier on, you did explain the fresh study that was trending earlier on this year, especially here in Southern Africa, or particularly South Africa to be specific. And now the question is, why is it such a big deal? Why is this a big moment when it comes to HIV cure research?
Okay, thank you for that question. This is so the reason the fresh study is a big deal is because, first of all, as I mentioned, those other studies of HIV cure, most of them have been using bone marrow transplants, which is, as I mentioned, is a very complicated, complex procedure. It's not one that can be rolled out very easily, because you require very specialized medical facilities, and it's very expensive to do. And it's also not very safe. So in the case of the fresh study, we wanted to try a different approach. And what we said is, what if we start antiretroviral treatment very so we had access to a cohort of women that we followed in KwaZulu-Natal, and we are able to detect when these women are in the acute phase of infection. So very, very early, within a few days of them getting infected, we are able to detect HIV infection. And then we put them on antiretroviral therapy immediately. So then we said, what about if we boosted the immune system, give them something that boosts the immune system, and then we stop the antiretroviral treatment? And that's exactly what we did. So we gave them the immune boosters that we gave them are two broadly neutralizing antibodies. Now, broadly neutralizing antibodies are proteins that directly inhibit HIV replication. So we gave them two broadly neutralizing antibodies. We also give them another that boosts the immune system called vasosalamide, or toll-like receptor 7 agonist. And then we asked those women to interrupt antiretroviral treatment. So this is the first time this kind of study was being done in Africa, and specifically in African women. Most of the other HIV cure studies have been done in the West, in Europe or America, and most of them have involved males, not females. This was the first time we were doing this in Africa, and also in women, and also the virus that is found here in Africa, particularly in Southern Africa, is a type of HIV that we call HIV subtype C, and it is different from the one that is found in the West. So because of all those reasons, our study is unique and shows that this kind of work can be done, and that this kind of strategy might be able to work among women in Africa living with HIV subtype C. So it's a significant advance forward for the field, and hopefully it will open the door for these kinds of studies to be done. So we are not there yet with the HIV cure, but it opens the door for many more studies of this nature, and hopefully that will help us to make progress.
Yes, and talking about doors, on our last episode, we were talking about HIV cure in children, and we learned that children have a special immunological features which make it's more likely that when it comes to HIV cure, we might develop an HIV cure for children way before other populations. So how is this a breakthrough?
Okay, so that's what we call a hypothesis. So we don't know for sure that that is going to be the case. But there is some indication that children, because they have a developing immune system, that the immune system develops, it might actually be able to take care of HIV or control HIV much better, particularly in children who start antiretroviral treatment very early. And so there is a lot of work going on to see whether children in particular, if they're given antiretroviral therapy early, and then that antiretroviral therapy is interrupted later on, are they able to control the virus on their own without the antiretroviral therapy? Indications that it actually might be easier in children because they have a developing immune system, and also there is some characteristics of the virus that infects children. So when children get infected, the virus that infects them might be different, might have some different properties from the virus that infects adults. And it would seem that that virus might be a little bit more susceptible to immune responses. And so this is why scientists are thinking that in addition to pursuing a cure in adults, it's important to pursue a cure or to do research to pursue a cure in children, because the children might be different, and this might then lead to that is suitable for children. So there's a lot of work going on in that space.
All right. And so our listeners, I think we did, when you were talking about the first study and also in children, how the studies are going, you talked a bit about treatment interruption. And I just want to just make it clear that these are controlled conditions, and, our listeners shouldn't say, oh, Professor Thumbi said that we can interrupt treatment, we can stop, and we will be cured, or I am undetectable, so it means that I can also stop. That they shouldn't. It's not a cure. ARVs are not a cure, and that these are studies that are controlled, and there's just, everything is regulated. You can't just decide to stop your medication and say, oh, I heard Professor Thumbi say that there's treatment interruption. Yes.
Oh, yeah. That's a very important point that you are making here, Sekabo. These kind of studies that I'm describing are done under very controlled conditions and under very strict observation by regulatory authorities and by clinicians, experts in HIV clinical management, who are able to closely follow the participants so that if something goes wrong, the participant can be put back on treatment immediately. So this is not something that anybody should try outside of a clinical trial, outside of a clinical study that is approved by an ethics committee. Nobody should interrupt treatment at home just on their own unless they are doing so in a clinical study that is by a regulatory body or an ethics committee. That's very, very important. It's very important that unless you're in that kind of study, you should always be on antiretroviral treatment.
Yes. And we're also talking about the different strategies or breakthroughs that have come when it comes to HIV cure research is gene editing. So can you please explain what gene editing is and how this might help people living with HIV in the future, if at all there is hope on that strategy?
Generally speaking, there are two ways of doing gene editing particularly when it comes to HIV cure. One form of gene editing targets the HIV virus itself. Because when somebody is on antiretroviral therapy, HIV has not disappeared from the body. HIV is hiding within immune cells. And so you can use some techniques to find out which cells contain HIV and then go into those cells and cut HIV out, remove it from those cells so that it is no longer there. So that's one way of gene therapy. That kind of gene therapy targets the virus. But there's another way of gene therapy, and the other way is to make the cells, immune cells in the body, much more potent, much more effective against HIV, so that they can attack the cells that contain HIV and destroy those cells so that HIV is. It may require you to take somebody's immune cells from their body and modify them in a way that makes them much more stronger or better at attacking the virus, the cells that contain the virus. So again, it can be a complicated procedure, but that kind of gene editing involves now what we call immunotherapy kind of gene therapy. So you are really helping the cells that fight HIV to be better at their job. So those are the two main ways of doing gene therapy for HIV cure. Either you go and target where HIV is hiding in the body and try to remove it by cutting out what is hiding within the genome, or you make the cells that fight HIV to be much better at that job, and there are certain modifications that you can do. But these are at the moment, these are experimental procedures. So we don't know exactly how to do this very well, but there are attempts, these so-called gene therapy attempts, and actually some of them are showing some great promise, even though none is has reached the market yet.
All right. And
About immunotherapy,
how does it work, and why is this exciting for HIV cure research?
Okay, immunotherapy tries to boost the immune response. So instead of trying to edit HIV out of cells, you give something, an immune molecule, that will be much better at fighting HIV. Now, immunotherapy is currently the most promising strategy, actually, for fighting and for curing HIV. And this is what we used in the fresh cohort. Because what we did was to give immune molecules, two broadly neutralizing antibodies, plus another immunomodulatory molecule. Now, we have a lot of experience. Scientists and clinicians have a lot of experience with immunotherapy, immune-based therapies for preventing diseases and even for treating diseases. And this is why, at this moment, these are the most promising strategies. They might, whether they will work eventually to completely cure HIV, we don't know, but they are at least showing promise in terms of controlling HIV for a long time without antiretroviral therapy. And so, because people have a lot of experience with them, with immunotherapies in general, even for other diseases, this is why there's so much promise that this actually might work.
Okay, and when we're talking about promises and also just seeing advancement of ARVs over the years, we know that from the start, people used to so many pills. For example, I'm a young person who's living openly with HIV, and I remember that there was a point where I was taking like 5, and then later on I went and took 2, and then now eventually I'm taking 1 pill a day. So we've seen advancement of ARVs over the years, and even now we're seeing that when it comes to prevention, also we're having long-acting injectables. So now the question is that are there any new drugs that are being tested right now that could bring us closer to an HIV cure?
Yeah, exactly. Yeah, and actually, I like the example that you've given about antiretroviral therapy and the way antiretroviral therapy has changed over the years. It used to be that in the past, people had to take very many drugs. They were very difficult to take, and then the, the drug regimens have been simplified and they become simpler. So for most people today, all they need to do is take 1 pill a day. And as you mentioned, there are even injectables now that you can take every 2 months in order to treat HIV. So in the same way that we have seen this evolution of antiretroviral drugs, we are hoping that when it comes to HIV cure, that maybe at the moment we can start by testing some very complex regimens. But the hope is that we will be able to move towards more simpler regimens and more effective regimens that are safer, and that we will, over time, be able to get to that time when we have to cure HIV, we will need only 1 shot, an injection to cure HIV. So we are not there yet, and we don't know how long it's going to take. It's a process. And in research, research can be quite unpredictable at times. Sometimes you make fast progress, sometimes you make slow progress. So, but the important thing is that you have to keep trying. And the important thing is that there are some very good ideas that are showing some promise. And so we need to keep advocating, and we need to keep doing this kind of research so that we can eventually get to a 1-shot HIV cure. But we are not there yet. We are moving towards that.
So we are towards a global HIV cure. And you did earlier on mention that when it comes to some of the, or how some of the guys who were cured of HIV, that they went through procedures that are very dangerous and that are risky. And also now you did give examples of gene editing or explain gene editing and immunotherapy. So with these, are there any challenges or risks that come with trying out these new treatments or procedures like gene editing and immunotherapy?
Yes, that's another very important point that you're making as a couple. These clinical studies are never without risk. There is always a bit of risk involved. We as scientists, and we try to minimize the risks that people who are in these trials go through. But sometimes you don't know for sure what will happen if you give a particular intervention, immunological intervention. We normally do a lot of testing before we go to a clinical trial. Before we test anything in human beings, we do a lot of testing to make sure that, to the best of our ability, the intervention is going to be safe. But you can never be 100% sure until you test that particular product in people. Yes, so there are some risks, but the risks generally we try to minimize, and generally they are low, but they do exist.
When now picking an approach, we did discuss that there are so many approaches like gene editing and immunotherapy, how do scientists now decide that, okay, so this is the best procedure that we're going to pick? Like, how do they decide that this is what we're going to test? And when it comes to now doing clinical or doing human trials, that this is the procedure that we're going to be using for this particular trial?
I think what scientists are trying to do is to provide options.
Okay.
Different options for people. So we test many different products that do different things so that people have an option. So, for example, for the people who are not living with HIV, the question is, how do I prevent myself from acquiring HIV? And so scientists have developed products, various products of various kinds to protect against HIV infection. But those products will not be useful for people who are already living with HIV. And so for people living with HIV, what the scientists have done is to develop medication that helps people to control the virus, control infection, prevents disease progression. But there are many products because not everything works in all the people. Sometimes some people will have some side effects if they take one drug, but not the other. So we need many drugs so that there is a choice, so that people can choose. If one drug doesn't work for you, then you have another drug that works for you. So the ultimate goal is to make sure that we have many products, but that the products are cheap or inexpensive as much as possible, and that they are simple to use, so that, for example, taking one pill a day is much better than having to take 3 pills a day. So these regimens so that they are easy for people to use, they are accessible, they are cheap, inexpensive, and available to as many people as possible, but also ensure that there is choice so that people can choose what works best for them.
All right. And when now we're talking about safety, how do researchers test that these new ideas are safe before now they reach people living with HIV when they're doing these clinical trials?
There's a lot of rigorous testing that goes into testing these products. So first of all, there's laboratory tests. You can test, scientists can grow cells, human cells, in the laboratory, and you can test your product in the lab to make sure that it is not possible to the cells, to cells that are from humans in the laboratory. But of course, testing in the laboratory is very different from testing in a living thing. So what scientists do is that after they test them in the lab, they also test them. Usually they will start with mice. They test the products in mice, and if there is no problem in mice, they may even go to other animals such as non-human primates, monkeys. They test the product there, and then they go into humans. And when they do the testing in humans, usually you start with a small number of people, let's say like about 20 people, and you test whether the product is safe, whether there are any side effects in the small number of people. And then now, if it's safe, the small number of people, you go to a bigger study, maybe involving about 200 participants. And then again, you get the safety data. And again, if the safety data is now okay there, that's when you now move on to test the big population. So everything done is done very meticulously, all the way from the lab to animal models to human studies. Initially, the human studies are very small numbers, and then you increase the numbers as you gather more and more evidence that the product is safe before you eventually roll it out.
And when now we're talking about these breakthroughs that are happening, especially here in Africa, like your study, the FRESH study, they they show that an HIV cure, or when we're doing these researches on HIV cure, that we must be inclusive and involve diverse communities. Why is this so important?
That's a really great question, Pablo. And I think the reason it's so important is because people living with HIV, they're diverse. Because HIV, it's very diverse in terms of the people that it affects. Then we also need to be diverse in terms of our approaches. HIV affects men and women. It affects people in rural communities, in urban areas. It affects children, adolescents, adults. So in the same way that all these communities and peoples are diverse, we need to cater for each one of them, because we don't want to develop something that works in one group but does not work in another. We don't want to just develop something that works in children but not in adults, or that works in adults but not in children. So it's very important that we think of all the groups that can benefit from an HIV cure, so that we consider them as we develop a solution, so that we develop a solution that targets everybody.
Okay, and now with like the big question, or what everyone wants to hear, is that we've been learning about all these breakthroughs that are happening around the world, and now we're seeing that Africa is now coming into the picture, and we're seeing all these breakthroughs that are happening when it comes to HIV cure research. So then the big question is, do these breakthroughs mean that an HIV cure is around the corner, or still need to brace ourselves and still hold on that there's still a long way to go before there is an HIV cure?
I'll be very honest and say that I think we need to brace ourselves. I don't want to give any false hope.
Yeah.
I do think that it's very important that we have made some progress, and that we are even having this conversation about an HIV cure, because many years ago, 15 years ago, we would not really be having this kind of conversation. But the fact that we have made some progress, and that the fact that we are beginning to see breakthroughs, like the one that we reported in a fresh study, gives me a lot of hope that with scientific innovation, with the creativity of young people, that we will be able to get to an HIV cure. But at the moment, we don't have one. So, and it's, as I mentioned earlier, it's very difficult to predict sometimes the pace of science. So we need to hold on a little bit, be patient, but continue putting in a lot of hard work so that we can move towards a cure.
All right. And then it is giving, hope. But obviously I know that people are like, no, there is a cure somewhere. We know it's there. They don't want to give us a cure. But also we're learning that, there are different procedures or different strategies that have worked for those that have been cured, and that they also had to go through very risky processes for them to actually be cured, and that it's not something that you can just wake up and say, oh, I want a bone marrow transplant, or I want this and that it's not something that's very scalable. So to those that are living with HIV, what can they learn from the success stories that you've had so far, like the London patient, Berlin patient, and recently the promising results from your study, the FRESH study?
I think what all these studies show is that it is possible to cure HIV.
Yeah.
And as it also shows that in order to make progress, it takes a lot of cooperation, multi-stakeholder cooperation. So many stakeholders coming together and cooperating and working together. For the FRESH study, for example, we would not have been able to do it without cooperation between communities, people in academia, as well as industry, and as well as government, because they are the ones who approve these kinds of studies. So we need everybody kind of working together, putting the best ideas on the table, and supporting this kind of work so that we can make progress. So I think what we can learn is that first, it is possible if we work at it. But secondly, that we need to work together, and we need to work very hard together in order to make progress. But I'm very optimistic that we can make progress and have even bigger breakthroughs than what we have seen so far.
All right. Thank you so much for making the time. Yes. And any last words?
Well, I always like to give a challenge to young people like yourself, Sikabo, because you are the future of our continent, and just say that there are so many, there's so much hope and optimism. And I think that we should be very proud that we have made a lot of progress in the fight against HIV. And there is a lot of space. There's a lot of work to do, but with the creativity and the energy and the intelligence of young people, we can make progress. So I work with a network called the Sub-Saharan African Network for TB and HIV Research Excellence, where we're trying to build capacity for HIV Cure Research. And there are many organizations like that, as well as advocacy organizations, and young people can get involved in this and make contributions, whether they are community activists, whether they are contributing through their working in research. All of us need to work together. It's not going to be easy, but we can. If we all work together, we will have much bigger breakthroughs, and an HIV cure can come from this continent.
Thank you so much. So an HIV cure needs or requires collective efforts. It's not just for the scientists or for the government to be pushing scientists and all of that. It's everyone. So we also need to be there. We need to be included, or we also need to step up and say that we're here in whatever ways that we can. Thank you so much, Professor Thumbi, and yeah.
Thank you so much, Sikabo. It's been a pleasure. I've really enjoyed chatting with you.
All right. Thank you so much to our listeners. So I'll see you guys in the next episode. Bye.